Our second speaker is Dr. Veronica Tom, a professor of neurobiology and anatomy at Drexel University. Her lab studies spinal cord injury with the goal of identifying potential therapeutic targets that will ultimately help restore function in people living with spinal cord injury. Dr. Tom has a neuroscience degree from New York University and received her PhD from Case Western University. Throughout her career, she has made innumerable contributions to our understanding of axon regeneration following injury that has been funded by several agencies, including the NIH, the Department of Defense, and the Craig H. Nielsen Foundation. Dr. Tom has received numerous fellowships and rewards, acknowledging her outstanding record of research, teaching, and mentorship, including the Julian Marsh Faculty Scholar Award from Drexel University, and her selection for the prestigious Hedwig von Amerigen Executive Leadership in Academic Medicine Fellowship. Please give a warm welcome to Dr. Tom as she takes the stage to give her talk entitled, Who's the Boss? Reflexes and Dysautonomia After Spinal Cord Injury.
Thanks very much. I can barely see over this podium. So thank you to the organizers for the invitation. This is a really cool opportunity to share some of the things that we are doing in the lab and as the spinal cord injury field, research field in general. So, okay. Oh, that's just another title slide. So a lot of what we think about when we think about the CNS and the brain and the spinal cord is thinking about really high level functions, right? So we think about things that make us uniquely human. So this is really nicely captured in this fantastic movie Inside Out, where you have the brain controlling various aspects of emotion, cognition, memory, learning. And those are all really important things to study and to consider. But there are other things that the CNS controls that's not as high level, right? So things like movement and sensation, critical things toward shaping our experience in the world, but not at essentially at the level of emotion or cognition. And when you think about things even more basic, the CNS also controls really fundamental functions of our entire physiology. So things like modulating blood pressure, immunity, kidney function. That's just to name a few. I'm just highlighting those because I'm going to touch upon them in my talk. So how is all of this possible? So we have to consider that the CNS is modulating the sympathetic nervous system. It's not quite sexy, but there you have it. And so we have, let me see if I can you see this mouse? No. Is there a way? Yeah. Oh, wait. Do you see it? Oh, okay. Oh, that's going to test my spatial skills. Okay. So if you look at, so these are all of these peripheral organs, effector organs that are innervated by the sympathetic nervous system. So they're innervated by neurons within the sympathetic chain that's out in the periphery, right? So this is the peripheral nervous system. But these neurons are modulated, directly synapsed upon by neurons within the spinal cord. And these neurons, I'll talk about the circuit a little bit more in more detail, but these neurons basically control a lot of the function of these neurons and drive innervation to these effector organs. Let me see if this works. Sorry, technical difficulties here. Okay, so what role does the CNS play in regulating sympathetic nervous system or the sympathetic function? And so kind of the classic model of understanding what a structure does is to know and see what happens after that structure has been lesioned or injured, right? And so we have something like that when we have a spinal cord injury. So spinal cord injury, when you think of someone with a spinal cord injury, you often think of someone who's in a chair. So the first thought probably is this person is paralyzed, and depending on the level of injury, that's the extent of the deficits, right? So lower level injury, you'll have paraplegic. Higher level injury, more of that descending input from the brain to the spinal cord is lost, and you'll be more likely to be quadriplegic, depending on the level of injury. But what's really important to consider is that the leading causes of mortality and morbidity are all related to things that are affected, sympathetic functions that are affected after a spinal cord injury. So you have susceptibility to infection. That's one of the leading causes of mortality, issues with circulatory system, as well as issues with the urogenital system. And we think that kind of key underlying causes for why these individuals are susceptible to these infections and to these disorders is because of a disrupted sympathetic nervous system. So if we go back to the schematic, we have all of these effector organs that are innervated by sympathetic neurons out in the periphery, but these neurons are regulated by neurons within the spinal cord. And so it's not hard to imagine that if you have an injury within the spinal cord, you're going to affect activity of these sympathetic neurons. And depending on the level of the injury will really impact how much disruption of the sympathetic system you have. So let's step back. How is that possible? So we'll step back a bit and think about the system as a circuit, as a really simple reflex circuit, right? So we're all familiar with, you tap the knee, that's a sensory stimulus that's coming into the spinal cord, synapses, and activates either one or two neurons. There are also some monosynaptic connections. And then you have these motor neurons that innervate the muscle. Those fire, and you get the leg kicking out, right? So it's a very simple reflex circuit that is really fast and importantly does not need the brain, right? You don't need the brain to see this reflex. So there's something that's very similar in the sympathetic system where you have neurons, you have input neurons, sensory information coming into the circuit that's relayed up to these output neurons that are, depending on the level of the cord, they're in kind of T1 through L1, L2. Those project out, they synapse upon sympathetic postganglionic neurons, which then innervate the effector organs. It's a lot of specifics, but basically the take home is that this motor reflex circuit is recapitulated or mimicked by the sympathetic reflex circuit. Sensory information comes in, output out to the periphery. In this case, instead of the muscle, we're talking about effector organs. So as I said, all of this, you don't need the brain to actually see these reflexes occur. However, the brain does modulate these reflexes. So there are inhibitory connections that basically tone down activity of the circuit, right? So what happens when you lose that circuit after a spinal cord, or you lose those modulatory projections after a spinal cord injury? Well, you lose a lot of this inhibition. And so what does that actually look like? So if you remember, the sensory information is stuff that's activating the circuit. If you give a sensory stimulus such as colorectal distension, which mimics constipation in individuals, which is a common problem with people with spinal cord injury, you see this dramatic activation of the circuit, right? This is basically now a hyperexcited, overactive reflex, and you'll get this really sudden and extreme increase in blood pressure because you have significant vasoconstriction, activation of the sympathetics, you get norepinephrine release, and that causes vasoconstriction. And so this is what, in real life, what heightened activation of this circuit means. You also get compensatory, most of the cases, compensatory bradycardia, but that's through the parasympathetic system that's intact. So if you look at, just to give you kind of a contrast, if you look at it, you give the same nasty stimulus, but to an animal, in this case, with either a lower level injury, so more of the descending input to this circuit is maintained, or an incomplete injury, so still more of this level descending input is maintained, you get a slight bump in blood pressure, but it's not nearly as dramatic. And that's because you have enough of this brain input to basically tell the circuit to chill out, right? So just as an analogy, if you have this kind of relatively infantile circuit below the spinal cord, this reflex circuit that is normally held in check by the parental unit, the brain, you have things that are it's a very happy, well-maintained, well-controlled situation. If you lose that parental unit, right, the spinal cord or the brain input down to the spinal cord, this circuit is now left on its own devices. And as you can see, it can wreak havoc, right? So this is a real picture of what my kids did when I turned my back when they were much younger. But that's essentially what's happening with the sympathetic circuit when you lose that modulatory input from the brain. Things are going a little bit haywire. So we have just this quick summary of what I've talked about. You have a high-level spinal cord injury. You lose that brain regulation of the sympathetic nervous system. That results in heightened sympathetic reflexes. That results in effector organ dysfunction. We talked about the cardiovascular system, right? So you have increase in blood pressure. I'm not talking about it here, but there's also vascular remodeling. That's not great. So the sympathetic nervous system innervates many organ systems. Think about that, one of the first slides that I showed you. So how does sympathetic hyperreflexia, these overactive sympathetic reflexes, impact other effector organs? So let's talk about another effector organ, the kidney. So this is kind of kidney for dummies, which is part of my speed. So the kidney is a highly vascularized organ, right? It's really important because it filters out all of this garbage out of your blood so that you basically have a nice, healthy, well-maintained homeostasis in blood. So you have unfiltered blood coming in, it gets filtered through the glomerulus and all the crap comes out, and then basically the blood gets recycled and all the garbage comes out of the kidneys through the urine, right? But because this is such a highly vascularized organ, you need to be able to maintain the amount of blood that's flowing through it. Okay, it's a really kind of neat intrinsic homeostatic mechanism to basically maintain kidney integrity. So because there's so much blood flowing through, it'd be highly susceptible to any damage through increases in blood pressure. So if you look, this is just a cartoon of a homeostatic process called autoregulation, where kind of in this physiological window of blood pressure, if you go increasing in the amount of blood pressure, you actually have more vasoconstriction. So that decreases the amount of blood that's flowing through the kidney to basically limit how much blood is going through the kidney at one time, right? To keep it at a steady state. So that's a really important mechanism to maintain the health of the kidney. So we looked at kidneys in animals that had a low level, so a thoracic level 10 spinal cord injury. So these animals do not experience sympathetic hyperreflexia. And you can see that when you look at the autoregulation response, that increasing changes in blood, so if you increase systemic blood pressure, you actually see a curve that's very similar to what I just showed you, where you have this dampening of, you have constriction to restrict blood flow that's going through the kidneys despite this increase in blood pressure. When we look at animals or kidneys from animals that have a high level of spinal cord injury, so these are the animals that experience that hyperreflexia, so those extreme periods of hypertension, you actually lose this homeostatic response. And so when you increase blood pressure, you don't have that vasoconstriction to limit blood flow, and blood is essentially pouring into the kidney. So remember, this is a protective mechanism to maintain the integrity of the kidney. So what does that mean? So this is in collaboration with a buddy of mine who's now at Case Western, and we looked at the kidneys in these animals. So this is a kidney of an uninjured animal. We stained for collagen, which is just connective tissue, but it's an indicator of fibrosis within the kidney or scarring. You can see that compared to a kidney from an uninjured animal, kidneys from these T3 spinal cord injured animals that experience these multiple bouts of hyperreflexia, sympathetic hyperreflexia, and these periods of extreme hypertension, and don't have that autoregulatory response, have a lot of scarring within the kidneys. And it's not something that's actually just specific to spinal cord injury. So if you have a low level injury where those animals don't experience these periods of sympathetic hyperreflexia, you see some collagen, but I didn't show the numbers, but this is significantly more scarring. So it's not an injury. It's not a peripheral response. This is something that is driven by damage to the central nervous system. So we talked about how spinal cord injury will affect both the vasculature as well as the kidneys. We're also going to think about the spleen, which is an important organ for mediating immune function, right? So if you look at animals, so this is all from rodents. This is a spleen from an uninjured animal, and you compare that to a spleen from an injured animal 8 weeks after injury. You can see that the spleen is from the high-level spinal cord injured animal. So these are animals that experience those repeated bouts of sympathetic hyperreflexia, right? That unchecked circuit. There's dramatic splenic atrophy. So you have the spleens are smaller, you have fewer splenocytes. And what does that mean in terms of immunity? So we wanted to directly test that. So we took animals, we squirted a little bit of Streptococcus pneumoniae into their lungs and looked just to see how well they did. So uninjured animals, when you give them that infection, they get sick, as indicated by a drop in body weight. So they weren't feeling great. But those animals recovered within a few days, and they got back to their baseline body weight, and all of them survived as long as we looked at, which is 10 days after the infection. When you have an animal with a high-level spinal cord injury, so these are the animals with that unchecked sympathetic circuit, right? And do the same infection challenge, see those animals get remarkably sicker. So they lose dramatically more body weight. And in fact, we actually had to euthanize a few of those animals because of the weight loss criteria that we set off, or that we put in our ICOOK protocol. When we pulled out the lungs from these animals after the infection, so this is 10 days after the infection, and cultured them on blood agar plates just to see how much residual bacteria was in the lungs. In the uninjured animals, there was some bacteria, but a lot of it was gone, especially when you compare it to an injured animal. So these are the animals that actually made it out the 10 days, that survived, but they're still pretty sick. So this is all of the residual. There's still a lot of residual bacteria within their lungs. So these animals, which have the splenic atrophy, that dysregulated sympathetic circuit, are not able to clear infections as well. So just to summarize a little bit, high-level spinal cord injury, you lose regulation of the sympathetic nervous system, get these heightened sympathetic reflexes that result in effector organ dysfunction. We talked about the vasculature, the spleen, as well as the kidney. So one thing that we have to think about is that we and others have found that over time, these heightened reflexes actually get worse. So these things are getting more severe. So it's not just loss of the descending input. That is an important cause for sympathetic hyperreflexia, but there are likely other factors that are at play. And so, we think that if we start to understand the mechanisms that actually underlie the development of sympathetic hyperreflexia, or these heightened sympathetic reflexes, irrespective of the loss of descending input, that may shed light on therapies that we can actually target to improve health for people living with a spinal cord injury. So one of the things that we started to think about was neuroinflammation. So neuroinflammation is associated with hyperexcited neural circuits, and it's been linked to other pathologies that are associated with hyperexcited circuits, and the degree of neuroinflammation correlates with pathology severity. So we looked at microglia, which are the resident immune cells within the spinal cord, and this is months after a spinal cord injury, and this is in tissue below the level of injury. You can see that the microglia are still highly reactive. Even in tissue, this is far remote to the injury. So if we have the injury at thoracic level 3, we've seen these activated microglia within lumbosacral spinal cord months out. And interestingly, we also looked at levels of soluble TNF, or tumor necrosis factor alpha, which is a master cytokine. It drives a variety of expression of a variety of different other cytokines, and that's also been linked to neuroplasticity. And when we look at this lumbar, or actually any tissue below the level of the spinal cord, even months out, we see elevated levels of TNF. So then we wanted to know, okay, we know, does inhibiting TNF, we have the players there. If we inhibit it, are we able to attenuate these hyperactive reflexes to improve and to diminish the detrimental consequences to effector organs? So this is just kind of a quick summary of a lot of work that a highly talented graduate student who's now in scientific consulting did. So if you look at kind of hypertensive responses in response to a noxious sensory stimulus to drive that reflex in injured animals that were treated with the vehicle, you can see that we get these dramatic increases in blood pressure, which is what I described before. If we continuously infuse a TNF inhibitor into the cord, or into the CSF, we actually, and in response to this nasty stimulus, you still see a response. So there's still some hypertension. So this is, I'm sorry, I should have mentioned, this is blood pressure. So this is millimeters of mercury. So this is all beat to beat blood pressure, and this is the mean arterial pressure, this line. You can see if you just compare the traces, this blood pressure trace to this blood pressure trace, you can see that the response is blunted, right? So there is hypertension, but it's not nearly as severe in the animals that were treated with the TNF inhibitor than the vehicle-treated animals. So we see diminished sympathetic hyperreflexia. How does that translate to the other effector organs? As I mentioned before, there's splenic atrophy after these high-level injuries where you have dysregulation of the sympathetic circuit. When we squirt in the TNF inhibitor, we see almost a normalization of splenic weight and more normal numbers of splenocytes within the spleen, which suggests that these animals have better immune systems. Does this actually translate into improved immunity? So we went back to that infection challenge that I described earlier, right? So those uninjured animals, they get sick. The injured animals that are controls get much sicker, and some of them have to be euthanized. There's a lot of lingering bacteria within their lungs. And when we look at the animals that were injured, but that were treated with this TNF inhibitor in the CSF, you can see that those animals got sick, right? So they still lost a lot of body weight, but remarkably, none of them had to be euthanized. They all survived till the 10-day post-infection monitoring period. And when we pulled out their lungs and put them on blood agar plates, we found that there is some residual bacteria, but it was dramatically less than what we saw in the injured controls. So this is just to summarize everything. So if you have a high-level spinal cord injury, you have enough disruption of brain input to this sympathetic circuit below in the spinal cord. There's neuroinflammation, probably other factors that we're starting to look at as well. That all results in heightened sympathetic reflexes, that results in effector organ dysfunction. And hopefully by targeting or understanding what these mechanisms are, that we can target them, we can improve, diminish some of these heightened sympathetic reflexes, and hopefully mitigate some of that dysfunction in the effector organs. So this is the most important slide. This is just a thank you for all the people who did the work. Some of them are not pictured. This was our first lab activity post-COVID when everyone felt comfortable again. So I'd like to thank basically everyone for being such a great group and letting us be as successful as we are. And then I'd also like to thank members of the Marion Murray Spinal Cord Research Center. It's a great group of collaborative faculty and investigators who are all focused on various aspects of spinal cord research, injury or not injured, as well as the funding agencies. Thank you.